Good morning, ladies and gentlemen, and welcome to our Executive Scientific Report series. Today, we will be looking at venom-derived pharmaceuticals, specifically 10 drugs based on venomous creatures that have been either FDA or EMA-approved or are advancing through clinical trials. These include a number of drugs that you may not have heard of before, as well as some that are better known. We will discuss their natural source, story of discovery, mechanism of action, current sales, and finally, look at the 5-year global sales outlook for each, taking into account any future approvals. Before we begin, if you are interested in getting access to our reports earlier than posted online and would like to receive them via email, please register your interest using the link below. Now let's get started. So what makes venom-derived molecules so attractive as a source of new pharmaceuticals? Many animals and microorganisms have evolved sophisticated weaponry to incapacitate prey, defend against predators, or compete for resources. Consequently, they produce bioactive compounds, including polypeptides, which are now recognized as potent tools for advancing therapeutics. These naturally occurring molecules exhibit high specificity and bioactivity, making them ideal candidates for drug development. Furthermore, many venoms consist of a complex cocktail of various toxins, offering a diverse chemical space to explore and diversify drug candidates. Moreover, the intricate mechanisms of these toxins have often co-evolved with their targets, resulting in high-affinity interactions useful in targeting specific disease pathways. Finally, considering the immense selective pressure that drives the production of venom components, these molecules tend to be highly stable, an essential quality for potential therapeutics. Over the past few decades, several venom-derived molecules have been successfully developed into FDA or EMA-approved therapeutics. Some examples include 𝗘𝗫𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏 Meanwhile, other venom-derived drugs such as 𝗘𝗫𝗏, 𝗓𝗏, 𝗔𝗡𝗏, 𝗘𝗫𝗣𝗟𝗏, and 𝗏 are continuing to advance through clinical pipelines. These compounds target a wide range of disease areas, from cardiovascular disorders to metabolic diseases and neuroprotection. Let's start with No. 1. 𝗔𝗡𝗜𝗢𝗢𝗏. 𝗔𝗡𝗜𝗢𝗢𝗏 is derived from the cone snail, a marine gastropod mollusk found in tropical and subtropical waters worldwide. There are about 800 species of cone snails, and they are named for the conical shell that they inhabit. Conotoxins are small proteins or peptides that act as neurotoxins, which the snails use to paralyze their prey. Cone snails inject their venom using a harpoon-like tooth. In the case of Conus magus, a species found in the Philippines, the venom contains a peptide known as 𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏. This peptide acts by blocking untype voltage-gated calcium channels in neurons. This mechanism inhibits the release of neurotransmitters like substance P and calcitonin gene-related peptide CGRP, which are crucial for transmitting pain signals. By blocking the release of these neurotransmitters, 𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏 effectively reduces pain perception. In the late 1980s, researchers began exploring the biodiversity of marine organisms near the Philippine reefs and realized that cone snails used a paralyzing venom to catch their prey. They isolated and identified a peptide called 𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏, which had the ability to selectively block pain-transmitting neurons. Through further research, it was found that this peptide interacted with a type of calcium channel called N-type voltage-gated calcium channels, which play a critical role in the transmission of nerve impulses. These channels are abundant in sensory neurons that carry pain signals to the brain. Further studies revealed that 𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏 binds to these N-type calcium channels, preventing them from functioning properly. This interference disrupted the release of neurotransmitters, effectively blocking the pain signals. Intrathecal administration of 𝗔𝗡𝗜𝗢𝗢𝗏 was well tolerated by patients even at high doses, indicating its favorable safety profile. Its onset of action was rapid, providing relief within minutes of administration. However, due to its requirement for intrathecal delivery, a procedure that involves injecting the drug into the cerebrospinal fluid surrounding the spinal cord. 𝗓𝗏𝗡𝗅 has limited use in treating neuropathic pain in ambulatory settings. But for severe chronic pain requiring continuous management, it remains a valuable option. Approval for 𝗓𝗏𝗡𝗢𝗡 was granted by the FDA in 2004, followed by the EMA in 2005. 𝗓𝗏𝗡𝗢𝗡 is currently marketed under the brand name Prialt. It is supplied as a lyophilized powder for reconstitution and injection. In terms of dosage, the recommended dose of 𝗓𝗏𝗡𝗢𝗡 is 10 micrograms administered intrathecally via a catheter placed in the lumbar subarachnoid space. Although it is difficult to estimate exact sales figures for prescription drugs, particularly those that are rarely used due to limited data availability, it is estimated that sales of 𝗓𝗏𝗡𝗢𝗡 currently generate around $30 to $40 million annually. Considering the limited patient population that can benefit from intrathecal therapy, it is expected that the annual sales will remain in this range over the next 5 years, totaling around $180 to $240 million between 2025 and 2030. No. 2. 𝗓𝗏𝗡𝗢𝗡 Bydureon. 𝗓𝗏𝗡𝗢𝗡 is a glucagon-like peptide-1 receptor agonist originally isolated from the venom of the Gila monster, a type of venomous lizard native to the southwestern United States and northern Mexico. The Gila monster is one of only 2 venomous lizards in the Americas and is known for its slow metabolism. While its venom contains no true anticoagulants, it possesses a unique property that helps regulate blood sugar levels. In the 1990s, researchers were intrigued by the observation that the Gila monster consumes very little food yet maintains stable glucose levels. They identified a peptide called 𝗓𝗓𝗓 as the active ingredient responsible for this effect. 𝗓𝗓𝗓 is structurally similar to a naturally occurring human hormone called glucagon-like peptide 1, or GLP-1, which is released after meals and triggers insulin secretion from pancreatic cells. Like GLP-1, 𝗓𝗓𝗓 activates specific receptors on pancreatic cells, leading to increased insulin production in response to rising glucose levels. Additionally, 𝗓𝗓𝗓 slows gastric emptying, giving time for postprandial glucose absorption and reducing hepatic glucose production. As a result, 𝗓𝗓𝗓 effectively manages post-meal glucose spikes and provides long-lasting glycemic control. It was initially marketed under the brand name Biotech and is now also available as Bydureon, a once-weekly formulation. Approval for 𝗓𝗏𝗡𝗢𝗡 was granted by the FDA in 2005 and the EMA in 2006. Currently, annual sales of 𝗓𝗏𝗡𝗢𝗡 exceed $1 billion. However, the GLP-1 market is highly competitive with numerous newer drugs available. Captopril is a medication used to treat high blood pressure. Its roots are pretty fascinating. It comes from the venom of the Brazilian pit viper. Back in the 60s, scientists found that certain peptides in the venom help lower blood pressure. Captopril works by blocking an enzyme that produces angiotensin II, a compound that tightens blood vessels. By doing this, it helps relax and widen those vessels, which lowers blood pressure. The FDA approved it in 1981, and now it's widely available as a generic. It's still a big player in the market, raking in about $150 million a year, and that's expected to stay steady for the next few years.